Majesty is the World Science Channel. Nice to see you there. Click the subscribe button so that when we upload the next video, you will be notified, and then endeavor to share this video to spread the light. In this video, I will be handling some sets of specimens in biology, and I'll tell you how to relate the specimens, the possible questions that might appear between the specimens. So we have in the list of the specimen the longitudinal section of fresh chili pepper fruits. We also have the mature fresh egg of catfish, the fresh egg of a domestic fowl. We have the longitudinal section of coconut fruit, ripe orange fruits, moist humus placed in a container, then dry humus also placed in a container. We also have a model of the uterus of any mammal containing a fetus there. Then longitudinal section of the ovary of Pride of Barbados flower, the leaf of Pride of Barbados, the panicum plant, which is scientific commonly called the guinea grass, then cocoyam plant, the caladium plant, then the comb of cocoyam, then the comb of cocoyam, so dry humus. These are the list of the specimens that I will be discussing in this video. You are advised to look at these specimens as if they are one. Try to relate them, find the differences and the similarities. Try to link one specimen to the other. It will help you. Now, we must not take them gradually. No. You will not take them individual. If you handle them individual without relating them, you will not get the possible predictions, because they are different specimens, but they are all biological specimens, which means there might be a link between them. So taking a look at the mature fresh egg of a catfish and the fresh egg of domestic fowl, there is a difference between them. At least the domestic fowl have shell, the thick shell, while that of catfish don't have thick shell. They have just the corine and then just zona pellucida sometimes, because of the reason that it is aquatic. It doesn't require protection from desiccation, but in that of domestic fowl, which the eggs are laid on the ground, terrestrial, so the shell actually protects the egg from drying up. That is, the shell prevents desiccation. So presence of thick shell in the domestic fowl, absence of thick shell in that of the catfish, or you can say a thin membrane of corine membrane present in catfish. That one is absent in the domestic fowl. But both of them exist at the cellular level of organization, provided they are not fertilized. They didn't tell us whether they are fertilized or not fertilized, but an unfertilized egg, no matter how big it is just a cell. Therefore, the biggest and the largest cell we have is the cell of the unfertilized egg of an ostrich. That is the Stratiocamillus egg. Then we have to also look at when they take a look at the uterus containing a fetus and the longitudinal section of the ovary of Pride of Barbados. If you look well, these two are reproductive in function. Similarities among them is that both possesses placenta. Yes, there is placenta in the longitudinal section of the ovary of Pride of Barbados. There is placenta in the uterus. But once you note this, that the placenta in the ovary is where the ovule are attached for Pride of Barbados, but the placenta in the uterus is where the fetus is attached by the means of umbilical cord. But what attaches the fetus to the placenta in the uterus is the umbilical cord, but the ovule is attached to the placenta of the ovary by means of a funicle. So there is funicle in that of longitudinal section of the Pride of Barbados. Then you should take a look at placentation. Placentation should be another topic you should focus here as far as this set of specimens are concerned. What is placentation? The pattern of arrangement of the ovule in the ovary of a flower, the pattern of arrangement of the ovule in the placenta of the ovary of a flower is called placentation. We have different types: the parietal placentation, the axial, the marginal placentation, the basal placentation, the free central placentation. So this longitudinal section of the Pride of Barbados have marginal placentation. That takes us down again. We have other longitudinal section like the pepper. The chili pepper have what we call the free central placentation. Some might say is axial. So then that of coconut have parietal placentation, parietal, the one we call parietal placentation. Then orange fruit though it's a whole fruit, you might be told to cut along the longitudinal section, have axial placentation. So these are the possible relationship between these specimens. Now, then, if you keep going down, we are required to produce the leaf of Pride of Barbados, then the panicum plant, that the Guinea grass, the whole plant, then the cocoyam and caladium. So one should be able to distinguish between monocotyledons and dicotyledons, especially in the leaf, the type of venation. So what type of venation do we have in the leaf of Pride of Barbados? Pride of Barbados, being a legume, is likely going to be a dicot. It's a dicotyledonous plant, therefore it will have network venation. But the leaves of the panicum, that is the Guinea grass, will have parallel venation because of what it has. It is a monocotyledonous plant, but dicotyledonous plants have a network venation. Then you might also be required to compare and contrast, or when we say compare and contrast, we are looking at similarities and differences. So what about cocoyam and the panicum, the Guinea grass? Both are monocots, both are monocotyledons, therefore they all possess parallel venation. Then taking a look at the panicum plant and then the cocoyam. The cocoyam and panicum plants, the cocoyam have underground stem called corm, which is another specimen, which is that CORM, corm, which is an underground vertical stem, as opposed by underground horizontal stem called rhizome. So, but there is no corm. Corm is absent in panicum plant, but corm is present in the cocoyam. In other words, there is no underground stem in the panicum plant, but both of them are monocotyledons. They all have fibrous root system. For similarities, presence of fibrous root system in the panicum and in the cocoyam. You should also endeavor to find their scientific names. Then the corm of cocoyam is a storage organ, of course. The storage organ there I've said it earlier that it is an underground vertical stem, corm. Yes, of course, that's that of cocoyam. Then beneltite is the roots. That reminds me, if you see cocoyam plant, the part above the soil are entirely the leaves. There is no stem. The stem is inside the soil. So that long thing that bears the blade of the leaf is the stalk of the leaf. So you can also compare and contrast between the cocoyam and the panicum plant by saying very long stalk present in cocoyam. The stalk in panicum is not long, reduced a bit. So then I'm talking about the corm. If they tell you, you can say it is a storage organ and it is also a perennating organ. You should know that cocoyam, of course, what it uses to move from one season to another season is what we call perennating. So that is to spend more than 2 seasons. It is the corm that we use to plant and it will give you new cocoyam, not the leaf. So the perennating organ there is the corm of cocoyam. Do you understand that now? Therefore, we now see humus and then moist. Yes, what is even humus? Humus is the organic content of a soil, the organic parts, which can be driven away by roasting soil in the air. And remember, soil rich in humus is the one advisable for agricultural purposes. So they might tell you which of the specimen is suitable for high crop yield. You say that of humus. Reason is because they have high content of organic matter, which includes the so-called fertilizer, so it will enable the growth of plant. Now, let's take a break and see this majestic periodic table. Then the clear answer to this analysis will follow immediately. The electronic configuration itself is all written starting from hydrogen down to the organesium, that one bearing atomic number of 118. That's up to 118. Then coming to the classification of the elements, this is just purely majestic. It indicated many groups and classes of elements that you never know they exist, and explained most trends. So it is purely a summarized chemistry textbook in form of a table. You see that classification you see there, you see the alkali earth metal, the alkali metals, the halogens, the chalcogens, indicating the common names of each group. apart from the one that is commonly known like the halogens, you have the all formers, the chalcogens, the nitrogen family. This table has come to answer your questions as a chemistry teacher, especially if you are in the class teaching. Your job is just to point at that table and the information you want will be there for you. Then this table do not only just contain the configuration, it shows the order, the half-bar order, how the electrons fill the orbitals. Why do we write 1s first? Then after 1s, we go to 2s, 2p. You see the arrow down indicating. And then it shows the number of shells present and the maximum electron each electron shell can hold. They are common named the K shell, the L, M, N, and also showed the formula for the maximum electron each shell can hold. So I cannot mention them all. Place order for your own copy of this majestic periodic table. You will not regret it. I've come to help you learn chemistry. I've come to help you teach your students very well. Present this. Can hang it everywhere. Remember, the periodic table, the Mendeleev table, is one of the most popular tables, and that's the table that represents science, even though it's chemistry. But anywhere you see that table hanging, automatically you know that this particular institution or this organization is science.
Hi, I wanted to tell you that you can contact Her Majesty if you want for your laboratory equipment and reagents. They are affordable and provide accurate results too. Her Majesty's word only sells what he knows, and his patterns of delivery are sure and certain. Place your orders today. Thanks for your patronage.
Since we have dry humus and wet humus, you can also go further to study how to know the percentage of water in a soil sample. You might be told that the weight of a wet soil is this, and the weight after dry is this. You just minus the dry weight from the wet soil weight, and then you get the mass of water. You now say the mass of water all over original mass, that is all over the mass of the wet sample times 100/1. So then if you come again, the next thing we should consider very well is classification of fruits. Definitely you will see that types of fruits. So we have about 3 succulent fruits that appeared in what I mentioned. Succulent or fleshy fruits. There are fruits actually that have a succulent pericarp. Of course, pericarp is the fruit wall. That is the combination of the exocarp, or what we call the epicarp, mesocarp, and the endocarp, collectively called pericarp, which is the fruit wall. So if you watch well, we have such fleshy fruit like the orange, ripe orange, and they said apart from ripe orange, we also have coconut. Is a fleshy fruit, it's not a dry fruit. Then pepper is also a fleshy fruit. So one should know the classes. To start with, all of them are fleshy fruit, but orange and pepper are all berry. But orange is a special berry called hesperidum. Hesperidum because it has a leathery rind. It has a leathery rind. Now, if you look at them again, the placentation differs, of course. I've told you that chili pepper have the free central placentation. Then the so-called coconut fruit is a drupe, D-R-U-P-E, meaning it has stony or hard endocarp. So you might be told to differentiate between the longitudinal section of coconut and longitudinal section of fresh chili pepper. You start from their placentation, you start from the nature of the fruit wall, meaning you can tell us that the chili pepper have a succulent endo. They have the endocarp, yes, the fleshy endocarp, while that of coconut have stony or hard endocarp. Then orange, when they say ripe, one can also look further to know the changes in plastid when fruits ripe. So the ripening of fruits can lead to the conversion of one plastid to another plastid, P-L-A-S-T-I-D. We have 3 plastids: the chloroplast, the chromoplast, and the leucoplast. So when the fruit is not yet ripe, probably we have the chloroplast. But when that is when it is greenish. But when it starts ripening, eventually you have the chloroplast change to chromoplast. So any other color you find around plants most times is chromoplast, but chloroplast is just the one that have the greenish pigment, which is chlorophyll. So as fruits ripe, the chloroplast content, the plastids might change. Then looking at this again, you should also know economic importance of all these specimens, provided mature fresh egg of a catfish, yes, can be used to fish fishery. Those people training, or I don't know how to say it, in fish farming, that's what we use to produce new one. And then looking at the egg, what connects the egg of catfish, the egg of domestic fowl, the ovary, and then the uterus, is that they are all connected with reproduction. Like I said earlier, the difference is that the ovary is an organ. That is, the ovary of the so-called pride of Barbados is an organ, while the uterus is also an organ. Then the fetus there is a complete organism. So an organism being inside an organ during development. Then you are also required to study very well the functions of placenta in the functions of placenta in a child during development. The placenta is the mediator, is the middleman. It acts as the lungs, the liver. It takes over the functions of the lungs until after birth. So anything that will get from the child to the mother and from the mother to the child must pass through the placenta, and therefore placenta is the mediator that connects the mother and the child. And remember that the blood of the mother and the blood of the child do not mix at all until childbirth, during the cutting of the umbilical cord. But during development, it is the placenta that causes the barrier in that case, such that it is through diffusion that materials from the maternal blood gets into the fetal blood. Do you get that now? Because if the maternal blood is allowed to get into the fetal blood, the pressure is too high, and the blood vessels of the so-called fetus is still tender. So the pressure, the force of flow might damage, not might, will damage the blood vessels of the fetus. So don't ignorantly say this child is inside me, therefore it has my blood. No, the fetus don't have, the blood of the fetus and the blood of the mother do not mix. If this is true, therefore the blood group of the mother will automatically be the blood group of the child. But it's not true. So a woman can have a child that has total blood, a blood group that is quite different from hers. So you just know, study well about development, also the functions of the uterus. The functions of the uterus, definitely it is the site of implantation. Then it holds and also nourishes the developing embryo until it becomes the fetus, until birth. Now they might tempt you to ask you what happens after fertilization in Pride of Barbados. The ovary, it turns into fruit. So you can also study about changes in the ovary of flower after fertilization. The ovule forms the seed. The ovule, O-V-U-L-E, forms the seed, while the ovary itself forms the fruit wall known as pericarp, just to stay up and doing in case of anyhow they ask the question. Then for the drawing of the specimen, you are advised to watch my video here on the rules, the basic rules for biological drawings. It covers all you need to know: how to label your drawing very well, the caption to get the maximum mark, because biological drawing is not just an artistic drawing where people will praise your handwork. So please check for that video and watch it. Search for the video under biology tutorials, WAEC practical guides, or NECO practical guide. Just do yourself good to enjoy what Her Majesty has been putting in place for you. My main aim is to give you the best, and that is what I'm doing. There are two ways to spread the light. One is to be the candle producing the light. Another is to be the mirror that reflects the light. Therefore, I am acting here for now. Anybody can be candle, but I'm acting as the candle, giving you this information. Can you be the mirror to reflect me by sharing this video? God bless you. I wish you the best of luck. Remain majestic for now. Don't forget to subscribe. Don't forget to keep sharing. Come back for testimony.